Hi, everyone. My name is Carolina González. I'm a fourth-year grad student working at Scripps Research Institute in San Diego. And today I will be presenting my thesis work explaining drug repositioning, a case-based reasoning graph neural network approach. And so I would like to first introduce my presentation today, saying that our work is motivated by the current challenge of developing new drugs. Drug discovery, it's an extremely long and expensive process that usually takes between 12 to 16 years and around to $1 billion to $2 billion to bring a drug to the market. And I think what is more concerning about developing new drugs is that usually the success rate to bring a drug to the market is really low. So as a solution, drug repositioning strategies take already existing drugs that have gone through some efficacy and safety evaluations and find new uses for them, which of course reduce the time and the cost of development. And there are certainly several experimental and computational approaches that are used for drug repositioning, but today I'll talk about graph-based approaches. And more specifically, I will focus on biomedical knowledge graphs. So biomedical knowledge graphs are a structured representation of complex biological data in the form of triplets, which each node in the graph represents a biological component, such as drugs, genes, proteins, pathways, diseases, and the biological relation that exists between these biological components is represented by an edge. And then the intuition of using biomedical knowledge graphs for drug repositioning is to analyze the well-known mechanisms of the drugs and try to find novel links with a novel disease. There are several knowledge graph link prediction approaches that have been already developed, where essentially the idea is to take the nodes in the input graph and try to map it to the embedding space in a way that the nodes that have similar local neighborhood will be close together in the embedding space. And then there is a scoring function that tells you how likely these nodes are together by a specific relation, in this case, the relation treats. And then, so this is great. Many knowledge graph link prediction models are already out there predicting good performance, the links between two nodes. But for drug repositioning and for other research areas in biology, we are not only interested in knowing that two drugs are connected, but we really want to know what are the biological mechanisms or the sequence of nodes and edges that explain the connection between them. And so taking that challenge in mind, we consider for this work developing a knowledge graph framework that allow us to identify the biological explanations for drug disease predictions. And so for that, we consider case-based reasoning paradigm. And case-based reasoning paradigm is an artificial intelligence framework that was developed many years ago, where the idea is to solve problems based on solutions that happened in the past. And I think a really nice analogy to understand this case-based reasoning paradigm is to consider a chef that is trying to replicate a specific tomato pasta dish, and in order for him to replicate this recipe, he needs to remember recipes that he did in the past and remember all the ingredients that he used, and then that way replicate the new recipe. So we can basically define case-based reasoning into 3 steps. First, given a problem, we first need to retrieve similar cases. And then once we set the similar cases, we reuse those solutions for our problem. And then finally, sometimes those solutions might not be very helpful for our problem, and so we need to revise to make sure that those solutions were helpful. So case-based reasoning for knowledge graph prediction was developed by Andrew McCallum Research Group in 2022 at UMass University. They use a subgraph strategy, and in their work, they use a semi-parametric strategy where they retrieve similar cases with no previous learning. They use a graph neural network approach to identify similar patterns between similar cases and a problem. This was specifically tested on a question answering task, and it was used for a specific benchmark data sets. But here in our work, we implement this case-based reasoning subgraph strategy for a drug repositioning framework where we don't only assess if the model is able to predict links, but also the ability to provide these interpretable biological mechanisms. So I will go into more details of how this work was done. And for that, I would like to explain it using a specific problem. So LXAI is a drug that is used for asthma. And so the question here is whether case-based reasoning model is able to identify this correct answer, but also their possible predictions. And so for that, the first step, as I mentioned, is to retrieve similar cases. And so here, what I'm showing is a set of similar drugs and the corresponding diseases they treat. I will go into more details of how exactly this works, but for now, we can notice that all the drugs that were retrieved here are also anti-inflammatory drugs for similar to LXAI. And so now the next step is to get the mechanisms of these similar cases that links every drug to the disease they treat, and extract the sequence of nodes and edges that explain this connection. And then use this sequence of nodes and edges to the problem that we're trying to solve. So LXAI, we basically explore the nodes around LXAI, and we are able to arrive to the correct answer, asthma, and as well to other possible predictions such as hypertension. Now, the important thing to notice here is that not all the solutions, as I was mentioning, are going to be helpful. So we see here that, for example, diarrhea and vomiting are not going to be the true solutions for LXAI. So therefore, the last step is to set up a revised step. And so taking in mind the idea of case-based reasoning, where similar cases should have similar solutions, we do graph neural network message passing framework to encode the local structure of the similar drug cases, as well as the local structure of the query that we're solving. And then we simply identify which representations are more similar between them. So as I was mentioning, to select the set of similar drugs, we need to calculate which one are more similar, right? And so for that, our model parameterizes each drug into a vector representation. And so this vector basically has the size of all the edge types that we have in our knowledge graph. And then an entry is set up to 1 if the outgoing relation exists for that particular drug, and 0 if it doesn't. And then we just simply calculate the inner product between all the drugs that we have and find the most similar one. So the intuition is that drugs that have similar biological relations should be similar between them, and we can reuse the solutions that they are using for our problem. So we wanted to test case-based reasoning for a biomedical knowledge graph. So we used a heterogeneous network that we built in our lab, mechanistic repositioning network, MIND, that captures almost 10 million edges and around 2,500 indications. We split it into the corresponding datasets. And we use hits at K metric to evaluate the performance of case-based reasoning to identify the correct indications, which basically just refers to the fraction of correct indications that are at K position or above. So these are the results. Case-based reasoning, we compare it to other knowledge graph completion frameworks that are also using a graph neural network strategy. And so we can see that they are all the metrics that we tested, case-based reasoning is outperforming them. And then we were also curious about the improvement of our model as more evidence is incorporated to the model. So what I'm showing you here is the improvement as more drugs are considered as similar entities. So we see that there is a drastic improvement from 1 to 5 number of drugs considered, and there is a slower rate of improvement, which just means that the model already has all the information required. All right, so I will just briefly go into a small case study that we were interested. So propranolol is an FDA-approved indication for hypertension, and it was later approved for essential tremor. So we were curious to investigate whether case-based reasoning was able to identify this link with our data set. And so what I'm showing you here is a PCA representation of the learned representations of the answer nodes for similar cases that is shown in red. And then all the other dots represents the predictions for propranolol. And I'm just highlighting the 5 top predictions to show that they are all clustered together with the answer nodes of the similar cases. And then, so as we see, the first prediction was hypertension as expected, and then we see Parkinson, which is a disease that is characterized by several motor symptoms, including essential tremor, which goes into what we were expecting to see with propranolol being repurposed for essential tremor. We carefully curated all the other predictions, the top 5 predictions, just to make sure that propranolol was actually studied for all of them. And then, as I was mentioning at the beginning, case-based reasoning has the ability to provide interpretable biological explanations. And for this, I'm showing propranolol and the top 3 predictions. So we see hypertension, Parkinson's disease, and thyroiditis. All the intermediate biological intermediates were carefully curated as well. But I think what is interesting to see over here is that the biological intermediates for hypertension, which is the approved indication, are also shared with all the other predictions that the model made. And so, yeah, I mean, for this work, we implemented a case-based reasoning model for drug repositioning task. We outperformed other link prediction frameworks. I think what's interesting to see that the model is improving as more evidence is incorporated. And what is interesting as well is that we were able to identify drugs that were already repurposed in the literature. And then finally, and I think most important, we are able to derive reasoning patterns from similar drug cases. And that's basically all I have. I just want to thank my advisor and the co-authors and collaborators of this project. If you have any other question, we'll be on the poster session.